Probably the most interesting and innovative feature of this meeting was how multidisciplinary it was, really a true breadth of people from clinical science, molecular genetic science, people who were working in animal model systems, people working in cell systems. It was really bringing this multidisciplinary group together that was the strength of the meeting.
Yeah, absolutely. And one of the things that struck me, and as a basic scientist, is that we had two first disclosures of clinical products here, and that's really unusual for this kind of meeting. That's usually very basic science focused. So that the breadth of it was extraordinary, and that was a tremendous aspect of it. And I think understanding at a molecular level, a lot of the physiological consequences of heart failure that we didn't know about, I think was really tremendous.
Yeah, and I, as a clinician, I really appreciated that everyone made an effort to bring that bench-side science right to the clinic, to where it may enhance our ability to precisely diagnose individuals and also tailor therapy. And that's where I see the future lies for making an impact in heart failure. And I got a real sense that at this meeting we had many talks where that potential began to appear.
Oh, absolutely. And then there was cell types that we haven't been paying too much attention to. The session on the immune system in the heart was really tremendous. There's so much potential there, and it's an emerging topic in heart failure, and it's something that I think has a lot of promise. And the way that it was addressed in terms of modern technologies of single-cell RNA-seq and cell depletion and the mouse models of heart failure, there's a tremendous breadth of insights there that are ripe for development.
And we learned so much about the latest ways in which all this data is being analyzed, the bioinformatic aspects of it, right? That's a field that's also evolving. And then, like this clinical problem, one of the reasons it's been so difficult to dissect is the lack of good in vitro models. And we had a whole workshop on that, and I thought that was a strength. You probably had more insights there than I did.
Yeah, it was really interesting because there was the breadth of in vitro models. They were all human stem cell-based models, but the breadth of them that included cell differentiation, bioengineering, genome engineering, and even sort of metabolic analyses of those models was really tremendous.
Yeah, and you said the magic word. I mean, for me, in my own particular area of research, I think having a session focused on metabolism, and that was, the partnered meeting right next door to us, the other Keystone Symposia, which is on the biology of exercise, had a lot of metabolism in it, and there was really good interactions, both people attending both sides of the divide, and also at the breaks and at dinner. We had lots of good dialogue, and I do see metabolism as another frontier. Almost every cellular process obviously is influenced by metabolism. And now we're beginning to have both the tools analytically and also to be able to manipulate, and we'll see where that leads us. There were several really excellent talks about clinicians who had firsthand experiences with patients and families and brought that passion of wanting to help that patient in front of them, and how they try to dissect the disease at the individual level, again, through molecular, then genetic, then cellular, then cell reprogramming, making whether it be myofibrils or cells or organoids from those patients, and then figuring out what the actual disease mechanism was, and then potentially optimizing drug therapy. And we had one, I thought, fascinating example of how a viral gene therapy
for a very specific disease.
Very specific disease, but for which there's no other cure. And it wasn't just one, we had several examples of that. Really inspiring part of the
I think it's clear that the combination of genetic stratification for various diseases, when possible, really enables the development of drugs or the diversification of drug regimens to various individuals. I mean, the concept of precision medicine has not been as well applied to heart failure as other diseases have been, but I think we saw a lot of examples of that in this meeting.
Yeah, and I really loved there were several talks where there was a historical perspective
Yes.
of where the field has come from, what lessons can be learned there, and how it might inform the future. And we also talked, I think, very intelligently about the challenges of, what works in vitro that may not work in vivo and why. And then when you move up the organism complexity chain into humans, where we need to be more precise in our preclinical work to be able to have fewer failures along the path of drug development into clinic. So that, as you said earlier, that intersection between having lots of industry representation, lots of clinician scientists, and the cutting edge basic scientists working in the field all together at one meeting was just phenomenal.
Yeah, I mean, certainly one of the things that we didn't have much of was human genetics, right? And that's something that, has worked for various types of diseases, but for heart failure is very, very hard. But I think with the new computational approaches that are coming along, that's going to be, I think, the next frontier for establishing personalized medicine programs.
Yeah.
With that said, I mean, there was a tremendous amount of single-cell RNA sequencing, for example, that was deployed, and the computational analysis behind that is extremely important to be able to make any sense of it. And when thinking about all the diverse cell types that we have in the heart, the fibroblasts, the myeloid cells,
Sure.
the endothelial cells, those.
And you'll recall we had one session specifically about the use of artificial intelligence.
Yes.
Right? So it was framed in the context of the enormous amount of clinical data that's now becoming available that might make tailored therapy, automate signals to patients through remote monitoring, telemedicine, but the back engine being an artificial intelligence type of system, whether it be machine learning, and then a lot of the bioinformatic analyses that you yourself are doing and others, are starting to deploy these algorithms. Things that the human mind can't grasp, that algorithms and sophisticated computational biology approaches are required. So I got several glimpses of that at this meeting.
You're absolutely right,
Yeah.
and those AI or machine learning, whatever you want to call them, really need to be able to go from patient records down to genetics with biomarkers in between.
Yeah.
And I think that's how we'll be able to understand such a complex disease as heart failure in a way that we never understood it before.
That's the hope.
Yep.
I think that's where, as a future direction, we need to sort of stay in touch with how that's progressing.
Yep.
How to fine tune that. Yeah.
Yes.
We had an entire session on fibrosis.
Yeah.
A key pathological mechanism of disease in variety of forms of heart failure, and just some beautiful talks about how fibrosis occurs, whether it can be modulated, are there different types of fibrosis. I mean, it was one example of but an emerging importance of fibrosis as a mechanism. And then I think coming back to what you talked about, which is the immune and inflammatory, another really important mechanism.
Yeah, I mean, we understand fibrosis, we understand hypertrophy. I think what is emerging and was really sort of a centerpiece of this meeting was cellular crosstalk. That all these cells are talking to one another, they're communicating to one another in ways that perhaps we had never anticipated.
Right.
And there's one talk, for example, that illustrated an actual physical connection of immune cells to cardiac myocytes that might be relevant to the physical properties of the heart. Nobody saw that coming.
Exactly. In other words, the heart's ability, we all knew to sense pressure, to mechanosense. We all thought it was related to the matrix and related to the heart muscle itself. And I agree with you. At this meeting, I learned that no, the immune cells are part of that as well. It's just beautiful.
Yep.
It makes perfect sense when you think about it.
Yeah, absolutely.
That all the cells in the heart are sensitive to mechanical forces. And as the geometry of the heart changes, both in physiological, with exercise, that we learned about in the other symposium, and in pathological, in disease, all of those cells are activated in unique ways and crosstalk.
Yeah. And there was a few interesting talks on you talked about fibrosis, like on the diversity of fibroblasts that exist in either the response to a heart failure regimen or in cardiac reprogramming, which is still an emerging frontier. And the surprise that there's such a diverse set of specific fibroblasts that are involved that have individual roles in both in those types of responses was really important.
certainly in almost every session, new targets were unveiled.
Yep.
And then I think there were a lot of really participatory industry representatives in the audience who asked important questions that I think would lead them and the investigators themselves to think about, is this druggable? Mm-hmm. Is this a target worth going after? And we had an entire talk about how you do drug development. Yep. So I think the answer to your question is yes, that many of the pathways that were discussed are potential avenues to look at. But there was a lot of humility and sobriety also generated by how careful one has to be and how thoughtful one has to be and how challenging it is to take a molecular target up through into clinical space.
Yeah, no, there's definitely a lot of molecular targets there. I think a lot of what was discussed was not just molecular targets, but how one might deliver to those particular cells and what might be involved in that. Yeah. And so the myriad complexity of drug development, I think, was illustrated at many levels throughout the meeting.
One of my favorite features, I mean, we touched on this at the very beginning, but I really liked it, and it was part of our planning as organizers, and I hope that Keystone continues to embrace it, because I've been to Keystone meetings before, which were very fundamental. Fantastic,
sure,
but very fundamental. And in this meeting, we tried in every session to have at least one sort of very more clinically oriented, and I think that really did what it was intended to do.
Yeah, and I think it's important for the basic scientists to be exposed to that sort of thing, because they're often not.
Yeah.
And I think it's for me, and for where I work, it's an institutional philosophy that we do basic science for a purpose.
Right, exactly.
We're always, even though we're studying molecules and cells, it's always to understand mechanisms of disease.
And that's something you and I share, is that I also work at a hospital research institute, and we have a large basic science group, but they know that they're there for the purposes of advancing care. Yeah, better diagnosis, better treatment.
That's right.
Yeah. As we begin to get all of the genomic data, proteomic data, physiologic data, and on the other side we have behavioral, socioeconomic, environmental influences. As reductionist basic scientists, we have to test hypothesis by hypothesis and test pathway by pathway, but several of the speakers brought to light this complexity that we need to figure out, maybe through computational biology, maybe through AI, machine learning. We need to figure out how we weigh what I would say the social, environmental, behavioral parts of heart failure. If you don't pay attention to salt and water restriction, if you don't exercise, I don't know that there's a gene for that.
That's right.
Right? And on the other hand, that there's a genetic predisposition. Clearly, there's immunological mechanisms, fibrotic mechanisms. Somewhere in that real tremendous complexity is the ability to say, for this patient, we need ABC, and for this patient, we need XYZ. That's the frontier. And it was brought up, the challenge is, unlike cancer, where it often is either a one or two hit that causes that particular cancer, and if you figure that out, we now have designer drugs for that. In heart failure, that's more challenging.
That's right.
That's the challenge.
And also, I mean, despite decades of understanding the pathophysiology of heart failure and the molecular mechanisms, we're realizing that the deeper we go, the less we know about this, and that there's broader sets of pathways that are brought into play. So that, for basic scientists, of course, it's a playground. There's more to learn, but it also means that if we think about it very carefully, we can understand new opportunities that didn't exist before.
Yeah. Yeah.
And hopefully some of those will be broadly applicable.
Yeah, absolutely. I mean, when I graduated from medical school, it was this disease, this sets of treatments.
Yep.
And now it's very clear that in heart failure, it's not one disease. It's one syndrome that presents very similarly in a clinical context, but it's probably dozens of diseases, which are all going to need their own individualized therapy. And that's another reason why meetings like this are great for clinician scientists, even some clinicians, is that begins, that, the curtain gets pulled. You get to see behind what's really going on.
And that's really exciting.
It's very exciting, yeah.
I mean, it was a fantastic meeting to organize. Mansoor approached me about a year and a half ago and said, let's do something really broad on heart failure. Let's bring in basic scientists, clinicians, industry, the whole thing. And that was instantly attractive to me. And the way that we put the program together was super fun. I mean, it was you know, we should do a topic on this. Who should we invite? And together we were able to come up with sort of a stellar cast.
Yeah, and I think, you know, the Keystone staff were great. They kept us on schedule. They were all busy people, but they really did a lot of the heavy lifting. But we had the privilege of thinking conceptually and scientifically, and then they did all the inviting. And I think, you know, I'll point out, I mean, I organize a fair number of meetings, I'm sure you do at your institution. Having the reputation of Keystone, both the, you know, the venue, how it's all done, the quality of the science, it never ceases to amaze me how easy it is to get the speakers we want to get.
Yep.
And that's what makes the meeting. So, you know, huge thanks from me to not only the Keystone staff, but to all the speakers who then let us have our vision. And then when you're here, it just runs so smoothly. But you're always a little nervous, you know, are people going to enjoy themselves? Is this thing that we, you know, imagined, this eclectic meeting, is it gonna work?
That's right.
So we'll wait to see what the evaluations say.
From what I heard, people are saying it works. So I'll stick to that.